When you're ready.
Should we experiment on the human genome? Yes, but we should wait until the technology becomes more advanced and we have a better insight into the field of research, which guarantees safer results and more successful results as well. We have to look through that. We have to look deeper into that question through the different various lenses, including the scientific, ethical, and economic lenses. First, we look deeper into the scientific lens.
Okay. So with the new and rising CRISPR technology, there comes its pros and cons with what comes with CRISPR. However, sometimes the pros can outweigh the cons. And with CRISPR technology, we're going to look at the positive effects first. With CRISPR, as we opened up the door with gene therapy, since we have more advanced technology. This can also help with children's health benefits, and this is hopefully the way to go. Not only does it help open the door for gene therapy, but also helps us get a better understanding with how infertility works and medical treatment options. So according to Ginkgo 2017, by providing a new way to study human development, Gremlin gene editing may lead to better treatments for infertility. Furthermore, Gremlin gene editing could be used to create cellular models and further our understanding of genetic disease. Such knowledge may be valuable in its own right, addition to leading treatments for serious diseases. The negative effects for CRISPR technology is off-target effects, which is we're going to be looking at first. So according to Cho, 2013, potential off-target effects in areas of the genome that are sensitive to double-strand break. And then for the last negative is effects on the immune response. So depending on the host's genome response, it targets plasmid DNA, but it could target either all the plasmid DNA or just parts of it. So according to the reason, it has been shown that plasmid DNA can also be inserted into both the on-target and off-target sites, and immune responses of the host can be induced by the bacterial sequences, which can dampen the performance of CRISPR. And how it can help sickle cell anemia. So sickle cell anemia affects 100,000 Americans and millions more worldwide. And scientists have known how to cure this disease since 1949, but they just didn't have the technology. Sickle cell anemia is a red blood cell disease which shortens the lifespan. And it's a hemoglobin protein sequence that is mutant. So it replaces the GAG sequence, which should be a GTG sequence. So on the studies, here is Victoria Gray, who participated in a sickle cell disease trial, and there's 31 patients that participated in this trial. All of them are healthy and are clear of sickle cell disease, and they all have very severe cases. So according to Steen 2023, doctors removed some of her bone marrow cells and genetically modify them with CRISPR and infuse billions of the modified cells back into the body. The genetic modification was designed to make the cells produce hemoglobin and hopes the cells would compensate for the defective hemoglobin that causes the diseases. So in this picture on the left, it shows Victoria and her treatment, and this is her now.
Now from the ethical lens. So as this technology becomes more prevalent in our society, so do questions about whether field research would be ethical. These questions have risen because there is no guarantee that research on the fetuses would be safe there may be much potential harm that may come to the embryo. So there's also a large question of informed consent on both the parents' end and the infant's end. This technology would affect the infant's entire life, and they have little to no say, well, they have no say in the matter due to the fact that they would be unborn while this technology is being administered to them. The National Human Genome Research Institute also reports a lack of knowledge that the parents may have regarding this technology that may lead to a lack of informed consent on research being done on their children and the risks involved. There are also various religious perspectives that we need to keep in mind. So the Islamic, Christian, and Jewish perspectives all share a viewpoint on, yes, this technology should be available for use, but only for therapeutic purposes. They also all agree that this technology would have to be strictly supervised, and we have to proceed with extreme caution when conducting this research. So there's also consensus that ongoing discussions between various forces such as religious leaders, scientists, and policymakers is vital in order to advance this technology into the future. And again, we have to emphasize on this technology being both safe and ethical for future generations. This CRISPR may also affect our environment in both the natural environment and the social environment. So, in the natural environment, gene drifting is a very likely possibility, and this would be defined as mutations spreading throughout generations over time, and these mutations may increase as the generations proceed through and throughout the future. This may lead to a lack of biodiversity in the environment, and natural imbalances will come from that. Native species may also decrease, and invasive species may also increase because of the subspecies having advantageous traits over the native species. And these negative traits that may be in certain species may also be transferred into other species. This technology may also create social imbalances and bias. So, because this technology may lead to people having physical and mental advantages, it may lead to social imbalances. And Brokowski has also determined this through a study in 2018. There may also be limited access to this technology due to financial availability, which leads to a new form of privilege in the world. And it may also be limited to certain regions of the world, which would deprive some of the human population from having access to the benefits of this technology.
Now we look at it at the economic lens. First, we have to look at the cost of the research. Here are some predictions from a report in 2018. Shows an increase in money toward the gene editing market. If those predictions come true, there will be an increase in research, which will again result in better results and safer and better results. On the other hand, these are just predictions, so it may not come true, which could again result in less research toward the gene editing, which would result in poor results and make it not safe. Next, we have to look at the cost of the treatment. Here are some examples of the cost. Zolgensma, which is a gene therapy for spinal muscular atrophy. Skyzono, which is for a gene therapy for a rare neurological disorder. And then Hemgenix, which is for gene therapy for hemophilia B. The high cost of these widens the gap between who has access to these, and the people with more money can afford these, but people with less money cannot. In an article in 2019, there was an example of a Louisiana Secretary of Health, Dr. Riccalagi, made a deal with a pharmaceutical company, Asegua, to treat people, or to pay off, and so that they could treat people. This is one example of recognition by research firms for the development needed to close the gap between who has access to these costs. Finally, will gene editing be beneficial or detrimental to the economy? First, the benefits are an increase in products and services, while also producing vaccines, like one for the COVID-19, therapeutics, and life-saving diagnostics. The negative effects could be that the negative effects of going deeper into the is harm toward people, the environment, and toward animals.
Okay, thank you. A little round of applause. Oliver, describe an argument from one of your peers' individual reports that made you think differently about your conclusion, the team's conclusion.
So the team's resolution makes me think about our individual work. Wait, will you mind repeating the question? Sorry.
Sure. Describe an argument from one of your peers' individual reports that made you think differently about your team's conclusion.
So, Juju's research made me more aware of the benefits in regards of, okay, the team's resolution made me think about it, and I'm lost, sorry. Would you mind repeating the question again?
Sure, yeah. Describe an argument from one of your peers' individual reports that made you think differently about your team's conclusion. All right, Oliver, you keep looking for that. Wes?
I found it.
Okay, you got it? Okay.
So the team's resolution makes me think about our individual work by letting me see the larger picture behind CRISPR and what it can do, and the cost behind it being extremely costly.
Okay, thank you. Wes, what is an example of a portion of a compelling argument from one of your peers' individual reports that you decided to exclude from the team presentation, and why?
We decided, one example is Oliver's on the testing of human embryos, because though, or we skipped over, on Gigi's report, we skipped over the in vivo applications since there was too much information, too much information for the presentation too.
Okay, thank you. Gigi, if you had another team member, what other perspective could they have researched that would have made a useful contribution to your project?
We looked at a futuristic lens, since it was researching more about research being done and what would be done regarding the gene editing, and also we would also do a political lens, as how it prevented the solution to gene editing, how has it been solved, like what are the beliefs behind it, and what are vendettas that could be.
Vendettas, I like that. Okay, another round of applause.